our dear colleague from Max Planck Institute of Evolutionary Anthropology, postdoctoral researcher Joscha Gretzinger, who is doing his research in paleogenomics, who started his way in archaeology, but fell in love with genetics. And, it just rolled out that, paleogenomics, paleoanthropology is the most important and impressive way to go. So let's listen what Joscha has to say about his research, how histories could be rewritten or complemented with the help of genetics research. So big round of applause and maybe some words from yourself.
I then finished my PhD, and now I'm currently a postdoc, partly in the HistoGenes project. And I will basically show you some results of that today. And this is, of course, hordes of barbarians, salvaged Germanic tribes that invade the Mediterranean civilizations, destroy their accomplishments and achievements for nothing. But of course, this is like a 19th century viewpoint on the history of the time period, and we have far proceeded from that. So clearly the historical tradition that starts basically already with Roman authors in that time and is then complemented by the interpretations of archaeologists and historians from the 19th and early 20th century. They propose there's this massive migration of mostly Germanic peoples from Northern Europe, from the territory of present-day Germany and Scandinavia to the Mediterranean, so southern and western Europe, France, the Iberian Peninsula, the Italian Peninsula, and the Balkan Peninsula, sometimes even into Greece. And later there are, of course, then migrations by other peoples, like, for example, the Slavs, the Avars, and so on. The social transformation is clearly impactful. We have the fall of the Roman Empire, or of course, this is a little bit coarsely defined here. It's clearly this separation into Western and the Eastern Roman Empire, and let's say in the end, the fall of the Western Roman Empire. Of course, the Eastern Roman Empire persisted much longer. What we also clearly know from the historical sources is the formation of Germanic kingdoms in Southern and Western Europe, the kingdom of the Franks, the Visigoths, the Ostrogoths, the Longobards, of course. And as we can trace it in archaeology, then the changes in material culture. We have changes in the burial customs, in the architecture, and even in subsistence strategies. So quite profound changes we can still see in the archaeological record. So the big questions are, of course, how much migration actually took place? Did homogeneous, closely biologically related groups migrate from discrete parts of Northern Europe, or were there heterogeneous confederations that were basically made up on the go, that collected more people the longer they migrated? And this is a very long-standing debate in historical scholarship. I think very mentionable here is the Austrian school of thought from the 1990s, especially Walter Pohl is very famous for that, who have questioned this traditional narrative of this big tribes of Germanic people that invade all these Southern European areas, and rather propose a more cultural acculturation process by heterogeneous groups that assert cultural dominance over the post-Roman populations. And the good thing is, of course, since we're here now in the medical campus, that natural sciences can help to answer these questions, because genetics, of course, is concerned with questions of ancestry and migration. So ancient DNA is able to resolve patterns of biological descent. And biological descent can be, of course, perceived in a short time period. That would be then biological relatedness that you see here on the left. That's basically just a pedigree of people, like everybody here in this room has that, with maybe siblings, with maybe children, but if everybody here has parents, that's logical. And these parents have parents, and it goes forth and forth. If we go even further back in time, of course, a person has quite a lot of ancestors. And that basically is then the transition from this short time period biological descent to long period biological descent or population genetics. And what we see here on the right is a phylogenetic tree of human populations built with a maximum likelihood approach based on allele frequencies, where you basically can also infer patterns of closer genetic relatedness, not only between individuals, but between groups. So genetics can reveal clearly some very important patterns. That's biological sex, it's biological kinship, and it's biogeographic origin of ancestry, which is not the same as the biogeographic origin of an individual. A very famous example that maybe a lot of you know is what you see here on the left, a principal component analysis, which is basically a genetic map. Each of these dots is an present-day individual or the genome of a present-day individual, and they're colored according to the region where they come from. So what you see here, all these blue dots that are individuals from the Iberian Peninsula, from France, Switzerland, Britain and Ireland, Scandinavia, the Italian Peninsula, the Balkan Peninsula, and Northeastern Europe. So what naturally emerges when we look at the genomes of these people, we see the geography of Europe. So you can reconstruct topological features of a geographical map from the genomes of people. So genetics is really powerful about origins of people. However, it cannot reveal social constructs. That's the cultural sex, so gender, non-biological kinship, for example, if you have like adopted children or siblings, the place of birth. I mean, you can be perfectly genetically like, I don't know, Lithuanian, but if your parents live in Paris and you are born there, genetics does not tell about that. And of course, your cultural identity and ethnic affiliation. If you're born as a Lithuanian or of Lithuanian parents in Paris, you probably still grow up as like a French person. So maybe you more identify as French than Lithuanian. And a very interesting example from the original 2008 Nature publication is this individual down here. It's a single person that is identified of Slovakian origin. So Slovakia plots here, but you know, all the other neighboring populations, Czech Republic, Hungary, they plot over here. Why should Slovak people so completely different than their close geographical neighbors? Now, of course, in this case, the self-identification of this person that was sampled here did not correspond with the genetic identity, because this individual is clearly of Southern Italian descent, but he or she identified as Slovakian. We don't know why, but it exists. So, to the HistoGenes project. The HistoGenes project is a very large and long-standing project. It was already conceptualized several years ago, and it will actually end next year, but there are many plans to continue it. There's many people involved here that are working on the integration of genetic, archaeological, and historical perspectives, mostly on Eastern Central Europe. That's basically what we mostly define as the Pannonian Basin. So Hungary, but also parts of Austria, of Slovakia, Czech Republic, Romania, and Croatia. It's led by 4 people, very, very, very important people. Johannes Krause, that's the tall guy here, my boss. Walter Pohl, as I mentioned him already, a historian. Patrick Geary from Princeton, also a historian, and Tivadar Vida from Budapest, who's an archaeologist. And they basically organized that over 6,000 samples, so genetic samples, should be taken. There's here this old number 500 for pathogens, but actually we screen all of the 6,000 samples for pathogens. And actually we were so successful in taking samples that and generating data that we not only have 6,000, but now in the end of 2024, even more than 7,000 samples. So it's probably, I would say, the largest ancient DNA project that currently exists. Of this 7,000 samples, at least 1,200 should go for isotope analysis, for reconstruction of mobility and diet, and we would generate at least 500 more C14 dates for the chronology. So the strategy is, as I already mentioned, that we try to be multidisciplinary here. So we have 3 major directions. We have the genetics that produces the ancient DNA data. We have the archaeology that tries to give us the context information, from which context the burials or the bones of these burials were retrieved, so that we know with what artifacts they were buried, in how the cemetery was organized. And of course, the anthropology was necessary for the age of the bird individual, the osteological sex, if there was some pathologies or trauma, to reconstruct a little bit of the life history of these individuals. And then we want to compare this with what we know from historical sources, so basically from contemporaneous authors that write about these events, Jordanes and so on, these people that describe these massive transformations. So a very important aspect here is the sampling of complete cemeteries. So that means we want to have in this region that I will show in just a few seconds to have as much data as possible and from different cemeteries. But if we select a cemetery, it should be fully excavated. We should have the complete archaeological documentation that we know exactly what is going on for each burial. And then we want to sample each individual. Of course, mostly not all individuals will provide ancient DNA. That's just the nature of ancient DNA. It's degraded, so it's not always possible to get from all skeletal material DNA. We were very successful with our current laboratory methods. We can retrieve from 98% of the sampled skeletal material ancient DNA usable for biological relatedness and population genetic analysis, so quite a large number. And that leads to the interesting result that if we have sampled a complete cemetery, we can, of course, understand how many people were related in the cemetery, how was the cemetery organized and structured. Do we see like a temporal pattern? Do we see patterns of biological relatedness, that people that are closer related to each other are also buried closer to each other or not? And we can, of course, understand how, for example, artifacts are giving down through generations. And if, for example, ethnic identities that are assigned via grave good assemblages like brooch types, swords, or whatever, actually are consistent with genetic origin. So we use methods that try to avoid biases. For example, we use genotype likelihoods and not only pseudo-haploid genotypes. That is more like expert knowledge. It's not so important for you. And that we try to produce these very large numbers. So normally we try to target cemeteries that have between 50 and 300 individuals. And in the context of that 7,000 samples that I spoke of earlier, we can really understand what happened in this area over this time. So this is basically an old map of all the sites that are going to be analyzed during that time period. What I want to highlight here is that, of course, most of the sites are in Hungary, but we also have quite a lot of material from like neighboring areas, especially here, Czech Republic, Slovakia, and Austria, and then Slovenia and Serbia and Romania, as well as a little bit from the potential emigration areas in like the more northwestern areas. What is also part of the truth is that roughly 3,000 of these samples are actually from the 7th century onwards, so actually early Middle Ages, so not migration period. But I think it's necessary to understand that as well. Why was Pannonia chosen? Of course, during 200 and 1200 CE, this area was inhabited by so many different groups that are described in the historical records. We have the Romans, of course. We have the Huns, the Sarmatians, the Gepids, the Langobards, the Avars, the Slavs, the Magyars. So there are so many different groups that it's very interesting to see how these groups interacted and how well these groups actually, the coming and the vanquishing of these groups actually correlate with changes in genetic ancestry. But we did not stop there. As I said, we have produced much more data, and that's basically where my job is currently mostly focused on. We have understood that only analyzing the immigration area where people come in is really difficult if we do not understand how the areas looked like genetically where they came from. So we produced much more data from Germany, from Czech Republic, and from Poland, even from Ukraine. And that's even not all sites that we're analyzing, but we're more and more expanding this for the future projects, that we really understand how these groups have moved. After skeletal material is taken, it is brought to either Brno or to Jena. This is now some impressions from Jena. That's our clean room laboratories. There the bones are prepared. You see, as typical for ancient DNA, there's quite a lot of safety measurements that we do not contaminate this very scarce genetic material with our abundant present-day genetic material. So we have to wear this overalls and multiple pairs of gloves and rubber boots and face masks and glasses, that we don't contaminate the samples with skin cells, hair, saliva or what else can like touch the bones. Then, of course, from the bone material, mostly from petrous bones or from teeth, because the petrous bones have the best genetic preservation. So this bone, it's part of the skull that has the best preservation of human genetic material, but it's a little bit less usable for the reconstruction of pathogens, because pathogens that are blood-borne are of course mostly found in bones that have quite a well blood support. So mostly teeth are very successful for these kinds of screening methods. And then when we have extracted the ancient DNA, it gets sequenced. In Leipzig, we have sequenced already more than 30,000 genomes, and they are then analyzed where I work here, this very nice institute, the Max Planck Institute in Leipzig, that was originally co-founded by Svante Pääbo, who got for his innovations in ancient DNA research also the Nobel Prize in 2022. Of course, there's also, as I said, the integration of archaeology. So we have a lot of data banks where we try to collect all this information that we have. This is such an Airtable. That's a software that we mainly use, where we have for each site, we have the individuals, and for each individual, we have the samples, so the skeletal material that was used from the burial, and then for the sample, then how many extracts and libraries were produced. And basically, there's many information given there. For example, we have GIS maps of the cemeteries that we can definitely always say which sample is located where in the cemetery. We can see which archaeologist is working on it. We have then lists where all the anthropological information is stored. The archaeological information was this individual buried, for example, with a shield buckle or with brooches, with what kind of brooches, how many pearls or whatever, how much different archaeological material. And this is then, of course, the invaluable resource that we can use to compare our genetic information with the archaeological information. And I just want to show you some impressions that can be used, or for example, how the usage of these different data types can be applied. So the first study I want to highlight here was published in December last year by my good friend and colleague Guido, and he analyzed a very large Avar period community from Raccocephalva. And what you can see here is a reconstructed pedigree from the genetic material that was reconstructed from that site. So what is possible is that to get from this 1,300-year-old human bone material to reconstruct a sixth-generation pedigree, where we can clearly understand who is related to whom, who is the father of who is the mother of who, and how this pedigree information is passed down from generation to generation. It has, of course, interesting implications, for example, for this spatial organization. For example, we can see here that in this part of the cemetery, there's a general trend that the earlier graves from the first generation are more in the east, and basically with proceeding time and proceeding generations, we see the cemetery was expanded to the west, and then the very youngest generations are then basically scattered around here. That's of course not the only information. It's not only just time, but for example, if we mark the nuclear families and the partners, so basically the parents of children as well as siblings and parents and offspring, we see that also individuals from different pedigrees are buried closer together, and that it's not only a temporal picture, but also individuals that are closer related to each other are also most of the time buried closer to each other. So on average, of course, it's not always like a perfect picture, but in general, closer relatives are also buried closer together. That's not, of course, completely unheard of. I mean, we do it the same often, but it's still interesting to see this in the archaeological record that we can actually show that. And of course, as I said, we can then compare the different artifact types. I'm sorry for the low resolution here, but for example, we can then understand how, for example, different grave goods were passed down from generations, and sometimes we see very interesting observations that, for example, the parents have a completely different cultural assemblage than their children, so that an ethnic identity has changed from one generation to the next. It's also very nice that we then can also to some extent reconstruct a little bit of the life histories of the people burned there, which is, I think, also nice, that we don't just see the bones in front of us, but also understand these were people, they had life, they had families. We have here this mother with her 2 children, a son and a daughter. The son died quite early, the daughter lived a little bit longer, but she had potential leprosy. That's then something that we can check in the ancient DNA record. So this is kind of interesting that we are able to get more insight into the life of these people. Another very interesting example that was published just last month, I was also involved in the study, is another Avar period cemetery. It's currently, Avars are really trendy right now, where we have analyzed 2 sites in Eastern Austria, very close to Vienna. One site called Mödling, and the other one is called Leobersdorf. It was part of the larger Avar cognate in East Central Europe. And the interesting thing is, so we have recovered nearly 600 samples from these 2 cemeteries, that they are archaeologically very similar. So they're basically both Avar sites, Avar cultural sites. But if we then looked into this 416 graves from Mödling and the 135 graves from Leobersdorf, we were rather shocked that they are genetically not at all the same. In blue, you see here Mödling, and these Mödling individuals plot in the genetic diversity of present-day Europeans. The Leobersdorf individuals, on the other hand, mostly do not overlap, with very few exceptions, with these Mödling individuals, but with present-day Central and East Asians. So the individuals in Leobersdorf are very well congruent with like an immigrating Avar core from East and Central Asia, while the Mödling population is more consistent with a local population that was acculturated, was assimilated in this elite, smaller elite group that came from the East. So that shows 2 very interesting things. It first shows that there was indeed, as I guess indicated by linguistics, by archaeology, and by the historical records, a long distance migration of people from Eastern Asia into the Pannonian basin, as it has happened several times with the Huns, for example, and the Mongols, but it's also now shown for the Avars. And we see that this Avar cognate was a multi-ethnic, heterogeneous political construct where people had the same material culture, maybe also the same language, we don't know, but genetically they were very different. And what we also see is they stayed also very different. There's not so much admixture. There's some individuals that fall in between, and since this is a genetic map and the distances between each dot is corresponding of genetic distances, then you can say that in many cases, if somebody falls between such 2 poles, they must be an admixture. So for example, these guys here are maybe 50% European-like and 50% East Asian-like. So there was a little bit of admixture, but overall, since we can reconstruct how the cemeteries were used over like 6-7 generations, not really that much. And what you can see here is the pedigree of the people of Leobersdorf, strictly patrilinear, and of Mödling, also strictly patrilinear. And Mödling is really amazing. There's 400 individuals, everybody's related there. So this is really a kin community graveyard where everybody is to some extent related to the next person. And this is just the largest pedigree. And I think it was in Vienna, the Vienna team printed it out. And if you have it on DIN A4 paper, it's like 5 meters. Like it's gigantic if you want to have it in a resolution that you can read it. So we can see that although they stayed there for quite some time, 6 generations, is quite some decades, they maintained a reproductive barrier between East Asian and European genetic material. We don't know why. That's not possible to say with genetics. We can just observe it. That's then something for archaeologists, historians, and social scientists. We can, of course, infer a little bit more about the patterns of social organization at these sites. What you see here on the left is the distribution of age-classified distribution of individuals, of males and females in Leobersdorf and Mödling. What you can already see is that in both sides, a little bit less in Leobersdorf than in Mödling, there's a significant surplus of male individuals. It's a very interesting pattern that females are missing. That's related to a practice that we see in many Avar period, but also Slavic period sites, that they are patrilocal and patrilinear. So the females come from other sites, and at a certain age, the females reach an age and have to go then to different sites where they probably marry than their partner. So there's always like a missingness of female individuals. We can also, of course, directly compare the inheritance patterns. This, like, it looks for patrilinear sites where we basically lacking the female counterparts of the parents. For matrilineage, we would be lacking the male parts. And if it would be bilingual, we would have seen a mixed pattern. And what we see is in Mödling, Rákóczi-Falva, and Leobersdorf, so in these three sites that I have presented, these other sites, is that in all of them, the vast majority of inheritance patterns is patrilinear. So the men stay at the site, and the son stays, and their son stays, and the females have to go. Or the other way, they come into the society. We can also, for example, compare then if the people were rather polygamous or stayed in their reproductive union. And what we see is that in Leobersdorf and Mödling, polygamous relationships are rather rare. We have a ratio of 3:1 and 4:1, but in Rákóczi-Falva, it was much more common. There it's 1.5:1. And that's very interesting because in Rákóczi-Falva, they have something called levirate marriage, that if on the male side, the female partner of one of your relatives, of your male relatives, the female your male relative dies, then you marry the female, so that basically the family stays safe and sound. And that is known from historical records, also from East Asia, so from the emigration areas of these Avar societies that is practiced here. And we can then trace these marriage patterns actually in the ancient DNA record. And the last thing I want to show from the study, which is also in my opinion very interesting, is that we can also measure long distance and very distant kin relationships. And then this is again how, like, the large scale data based on a larger geographical area is important. So this is again all different sites from the Avar period that we have analyzed. And these connection lines are distant kin relationships that we can detect with ancient DNA. And the broader they are, the thicker they are, the more kin connections there are. And what we see is that all these Avar period sites in the Avar cognate or territory of this former territory are very well connected. So what we see is that these are not isolated places. These are actually very well connected centers where these communities exchange. And this is of course necessary. If you're pottery local, then you need a good network where you send your women away, or where you get your women from. And that's probably, and that's also what we actually find, it's not in that study, that we sometimes find the female daughter, the missing daughters or the missing mothers, like in other sites, sometimes hundreds of kilometers away. So I hope that gave us a little bit of an insight how HistoGenes works and how it can produce very interesting insights, not only for geneticists, but also for archaeologists and historians and maybe also linguists. And that although the project is just starting to publish all this interesting material, that it's already pretty successful in publishing high impact papers. However, my interest is less in the Pannonian basin, but more to the north. And that's what I want to highlight now. This is a study I'm first authoring, and it's currently in Nature and Revision, so it will be hopefully out in summer this year, and it's about Slavic migration. That's of course the current political climate, a very sensitive topic. I try to be as uncontroversial as possible. I think what we cannot deny is that the Slavic language family is the largest Indo-European language family in Europe. There are roughly 350 million speakers, mostly, of course, in the east of Europe, but also in the southeast of Europe. It's quite distinctive distribution. And traditionally, again, there's a traditional view how that came to be, is that they connect this to a migration of Slavic people, so people that spoke Slavic languages from different areas in Eastern or Central Europe that expanded across this geographical area and brought the Slavic languages. So there are three major concepts where they came from. There's basically the migration hypothesis that locates the origin of these Slavic-speaking groups in present-day Ukraine and southern Belarus, and from there they went then west and east. Then there's the model that was for a long time favored by Polish archaeologists, or at least some of them, that basically presumes a Slavic continuity in Poland at least since the Iron Age. And then there's the hypothesis that actually the Slavs are more like a concept made up by the Byzantines, and they are actually not a real uniform ethnic group, but more like a complex confederation of different groups. What we also know is that it's clearly not just a linguistic phenomenon. We see during the time very similar archaeological ceramics. This is just what like archaeologists tell me, because I'm not so well acquainted with the with ceramics. So I don't want to hurt any feelings here, but they say it's rather simple compared to often the material culture that you find in these areas before the Slavic period. That's what Mario Slaus told me. He's Croatian, so I hope it's not like I'm not saying this is like very bad material. That's what they tell me. And that connects all these areas. So there's clearly also some kind of a material transformation, not only a linguistic transformation. And to potentially give some hints, some possible explanations, we have started a genetic project that is a little bit different from the general HistoGenes project. That's because we like have, especially from the more northern areas, smaller but more diverse sites. And we thought, okay, we can simply try to see if we see in these areas where we have this archaeological and linguistic transformation, also genetic change. So we have created 3 different sampling transects from Central Europe, one in Eastern Germany, one in Poland and the northwestern Ukraine, and in the northwestern Balkan, which is mostly Slovenia, Croatia, and some parts to the east. And for the northwestern Balkan, we have roughly created 100 genomes. Most of them date shortly after the transition to Slavic material culture to the 7th and 8th century, but we have also some Roman period material from that region. Then we have created 330 genomes from Eastern Germany, from the Thuringian time period, so that's the 5th and 6th century, or the first half of the 6th century, as well then from the 9th and 10th century, as well as some genomes that date a little bit earlier to the Iron Age, as well as, I'm very proud of that, material from Poland and Ukraine, mostly material that dates to the High Middle Ages, but also probably what I was told the probably oldest inhumation burials that are currently securely dated, that date to the 7th century, 7th and 8th century from the border of Poland and Belarus, as well as some genomes from the Roman Iron Age, as well as one early Iron Age genome. So in total, we have 600 genomes, and we can now see if something happens in these regions. So again, this is such a PCA plot. This is a high-resolution data set I have built with data from many different projects. Here are the Lithuanians that I now learned were produced here at these institutions. So very nice to connect now finally some faces with this data. And what you can see here, it follows again a little bit geography. It's not as nice as the PopRes plot shown earlier, but here you see Britain and Ireland, Belgium, France, Iberian Peninsula, Italian Peninsula, Sardinia. Here you have Scandinavia. Germany built such a bridge between West and East, the Baltics, Finland, East Central Europe, and the Balkans. Okay. What we can do now is we can look how the populations in these areas have looked genetically before the Slavic period. So I just arbitrarily now define this here as roughly 200 to 600 CE. This is just for simplification. And what you see here in the northwestern Balkan, so from Croatia and Slovenia, individuals look very much like present-day individuals from Italy, or maybe like also from Spain. For Eastern Germany, that's also not so surprising. They pretty much plot with present-day Dutch, Danish, and Northern Germans, but there's also some interesting diversity to the south. So there was a lot of exchange seemingly with the Roman Empire and the remnants of the Roman Empire. And for Poland and northwestern Ukraine, we see that they're actually not genetically so different from the Eastern German samples. They plot on present days in Norwegians and Swedish. And those here in the room that are a little bit familiar with, like, for example, Jordanes, he's a Roman Gothic author, and he wrote that the Goths that are often equated with the Wilbar culture actually migrated from Skansa, from Scandinavia, to Gothic Skansa, which is northern Poland. And what we see here is that genetically, the data is very, very congruent with such an idea, that they look very similar to both ancient and present-day Scandinavians. So we have a very diverse genetic makeup during that time, before the Slavic period. After the Slavic period, this changes, or with the Slavic period. What you see here for Croatia and Slovenia, we have this massive shift in the direction of present-day Northeastern European populations and Eastern European populations. So from here to here, they're pretty much plotting already together with present-day Croatians, very close to Polish, that would be here, and Lithuanians would be here, and Belarusians would be here. So there's clearly a massive genetic shift into the direction of Northeastern Europe. The same thing in Eastern Germany, complete shift from the west to the east. And most interestingly, of course, for the question of population continuity in Poland, we have exactly the same pattern in Poland and Ukraine, that the former gene pool, that's more like Northwestern European, disappears, and the Slavic periods, so 9th, 10th century, and even earlier samples from the 7th and 8th century, plot pretty much on top of present-day Polish individuals. So yeah, we can be pretty sure that there was a massive genetic exchange. So if you would compare this all in one picture, we see we have, during the Roman and late antiquity, we have a very diverse set of different ancestries, and with the transition to the Slavic period, we have a homogenization, and basically during that time, genetically speaking, individuals from Eastern Germany, from Poland, from Ukraine, and Croatia and Slovenia look genetically very similar. So over hundreds of kilometers, a very similar genetic profile. We can even put this in numbers. For example, for Eastern Germany, we calculate that the genetic exchange of ancestry or replacement is roughly 85%. For Poland, it's 93%, and for Croatia, it's 80%. So in all cases, over 80% of the genetic material of the pre-Slavic times are replaced by ancestry from Eastern Europe. This is just another visualization how that could have looked like. This is again pre-600 CE. In blue is like Northwestern European ancestry. This very small map you see down here, that's basically ancestry that was distributed during the Iron Age and the Late Bronze Age in these areas. And in purple, it's Eastern or Northeastern European ancestry that's basically mostly distributed in the Baltics. And what you see here, there's basically it exists in lower fractions basically everywhere, but really high fractions only exist during that time in Lithuania as well in central Ukraine. But west of that, in Poland, as well here in the Svilbar culture sites from, for example, Grodek, as well as from Cherniakov sites in Ukraine, we see mostly Northwestern European ancestry. And then after 600 CE with the Slavic period, basically in all sites, we see this Baltic-related Northeastern European genetic component, and this 80 to 90% genetic replacement. And interestingly, this replacement is without sex bias. So males and females from Eastern Europe come to the West and to the South, admix, replace the gene pool, and create basically the genetic landscape that we see to some extent still today. That we basically have this current genetic distribution of Eastern European ancestry, even in Czech Republic and Poland. That is, of course, now in comparison, for example, to modern DNA data. We can also do this with ancient DNA data. This is a very cool method that's called IBD, identity by descent. That's very common in population genetics of present-day data. It's very hard to use it in ancient DNA data, or it was very hard because you need diploid genotypes. I don't go into detail here, but in theory, it works like this. I mean, you always you get always like inherited parts of your DNA from your parents, and your parents are again just a recombination of the genetic material of the chromosomes of the grandparents. So you're basically just a mixture of the genetic material of your grandparents. You see it in this case. There's this grandfather here, and he has this orange chromosome, and he has two children, and both his children, his daughter and his son, inherit fractions of this chromosome. It's recombined with his blue chromosome. So they already have just a fraction of their ancestry left from their father, and then they have partners and their own and create children. And then here, this individual this daughter has a little bit of this orange ancestry from her mother, which is ultimately derived from her grandfather, and the son has a son with this female, and also inherits part of this orange chromosome from the grandfather. So although they are not longer close, I mean, they stay closely related in the schema, but over the long time period, although these individuals are not direct siblings or parent offspring, they share DNA parts of the chromosome that are identical because they derive it from a common ancestor, from this grandfather. And we can use this technique to detect very long, distant relatives. And that means, I would say, until the 10th degree. And I mean, for most people in this room, 10th degree means nothing. We would not identify 10th degree as a relative of us. Probably even I would have 10th degree relatives in this room, although probably they go back 300 years. That's very common in Europe. And what we see if we compare this measurement of relatedness in MP, so migration period sites, so pre-Slavic sites, and SP, Slavic period sites, we see that the MP sites from Eastern Germany, from Poland, as well as early medieval sites from Northern Germany and England, so areas that don't experience Slavic migration, they share more distant relatedness with each other, while the Slavic period sites share substantially more distant relatedness with each other. And it's very interesting that for example, a person from 5th century or 6th century Eastern Germany shares more relatives with a contemporary person from Poland, from 100 kilometers to the east, than with the person living in the same village just 200 years later. So basically, you can, during like the 9th century, you can go to Ukraine, take an individual, and there's quite a good chance that this individual will have a 4th degree relative, and 4th degree is much closer in Ukraine or in Poland, because these people were so closely related to each other. And just again, that we also can again reconstruct something about the life histories of these individuals. We have used this whole symmetry approach in Eastern Germany. I want to highlight one site. It's called Niederwünsch. It's in the south of Saxony-Anhalt in Eastern Germany. It's 200 individuals, and we have reconstructed several pedigrees. Here's a very large one, as well as a smaller one, and then some core families. It's very interesting because we have from the same area also this migration period sites, and what we see in the migration period, nearly nobody is related. So if you compare all pairs in a migration period site from Eastern Germany, only 1% of all the pairs show distant relatedness. In Niederwünsch, 20% of all pairs of individuals, so if you just compare this individual and this individual and this individual and this individual, will be distant relatives, 20%. So basically nearly everybody is to some extent related to each other. That's a massive increase of relatedness. We can also see that, again, as in Rakutsifalva, that the relatedness is reflected in the organization of the pedigree. So here this would be the old church, so they were already Christianized, and people that are closer related to each other are also buried closer to each other. There's like the red pedigree, there's the orange part of this pedigree, there's the blue part of this pedigree. So these people were, they wanted to be buried closer to their relatives than to strangers. What we see is, for example, that the people that are closest buried to each other are actually daughters and their parents, so mothers and daughters and father and daughters, which is very interesting because generally we're lacking the daughters. Because just as with the other sites, if we look at the individuals that have no close relationships, here for the subadults and for the adults, there are much more females that have no close genetic relationships, because often in this case it were probably individuals that came from different regions, probably to marry a local man and did not reproduce until they died. So we have a surplus of non-related females, but generally we are lacking females. They are missing from the pedigrees because we just have the males. You can see it here. I've colored all the patrilinear patterns in blue and the matrilinear patterns in red. So what we see is 90%, roughly speaking, of all the inheritance patterns we see in Niederwünsch are patrilinear. So we have a patrilinear society here. We also see it in the direct comparison of close relatives. So close relative, I define everything closer than 4th degree. We see on average or on median, females have roughly 4 close relatives at the site, while males have nearly 9 to 10 individuals, and the spread is also much higher. There's some males that have 25 close relatives at the site. Also, the individuals that have absolutely no relatives at the site is very different. For males, it's only 6. For females, it's 23. So it's clearly there's also a signal of patrilocality here. Also seen, for example, in markers of genetic diversity. this is the Y DNA. The Y DNA is always inherited from the father to the son. mtDNA is inherited from the mother to the children, not only to the daughters, but also to the sons. But basically, they represent the patrilinear and the matrilinear inheritance. What we see is that the diversity of mitochondrial lineages is very large, while the diversity of Y chromosomes of patrilinear lineages is super small. Basically, all the males are related to each other. They have this very interesting haplogroup R1a. It's derived from the corded ware culture. And it's very indicative because this is still like the most common Y chromosome haplogroup all across like Slavic speaking or Balto-Slavic speaking Europe, and even in non-Slavic speaking areas of Southeastern Europe. So one could maybe think that such patterns of patrilocality, of patrilinearity can of course create such population genetic patterns if males with this Y chromosome lineages are somehow advantaged or over-advantaged in reproduction. On the other hand, if we compare then, for example, the distant kin relationships, we see as with the close relationships, which is shown here as pie charts, there are much more close male-to-male distant kin relationships within the Slavic period sites. This is Niederwünsch, this is another site called Steuden, it's a site called Obermöllern. And within sites, males are closer related to each other. But if we compare between sites, and it's the thickness of these lines here and the percentages, between sites, the females are closer related, because basically the relatives of the females that we don't find within the sites are in the other sites. This is basically a smoking gun for female exorgamy, the practice to send females away from marriage. And that produces the interesting pattern not only in Eastern Germany, but also, for example, in Poland and Ukraine, that the migration period and Iron Age sites have very strong, high Y chromosome diversity and mtDNA diversity because they are not strictly patrilocal and patrilinear, while the Slavic period sites have very high mtDNA diversity, but very low Y chromosome diversity because the males always stay within the sites. In terms of population genetics, we can do this IBD measuring between the ancient population and present-day Europeans to see who's today sharing the most common ancestors with this Slavic individuals from Eastern Germany, from Croatia and Poland. And it's actually Latvians and Lithuanians. It's very interesting. It's not Polish or Ukrainians. It's here the Baltic populations, also Belarusians actually. But that also translates into prehistory. So if you look at all samples from Europe that post-date 3000 BC, so basically after the Neolithic, and we interpolate the amount of shared drift, which is a measurement of genetic affinity, we see the populations that are closest related to Slavic period individuals from Eastern Germany, from Poland and Croatia, are Bronze Age and Iron Age populations from the Baltics, from Lithuania up to Ingria. And we can use this information. We can use the geographical information of the ancient individuals, the temporal information, so how they date, and the affinities to interpolate that to predict where probably this ancestry came from. This is a very cool method developed by one of my friends and colleagues, Clemens Schmid, that uses uses all this information to predict where an individual came from. And this is basically for these different Slavic period sites from Eastern Germany, from Poland, and from Croatia. Velim is a site in Croatia. And that predicts where these individuals have lived at 100 CE, so before the Slavic migration. And for all the sites, it's very similar. It's in close vicinity of the Baltics, somewhere in the area between northern Ukraine and Belarus. Unfortunately, as you maybe have seen from this map, this is exactly the space where absolutely no ancient DNA samples are available from. Yeah, that's a little bit of an issue. So currently, our idea is, and this is of course not a final verdict, is this gene pool, the Slavic gene pool has formed as a rather complex admixture. We have like a Baltic Bronze Age derived source that inhabits most of Northeastern Europe, that expands southwards into the area of maybe southern Belarus, definitely central and maybe even western Ukraine. It mixes there with southern European ancestry that's Balkan related and forms the gene pool. And maybe it gets a little bit of ancestry from like northwestern European ancestry that's like in Scandinavia, northern Germany, that comes from the west. And from there it spreads then both east and west and contributes ancestry to present-day populations. And that's basically the amount of Slavic period ancestry. So actually the ancestry that we have observed in this earliest inhumation births from Poland, how much present-day populations have that, and we see the highest amounts are still, of course, in Ukraine, in Poland, and in parts of Russia. And then the further you get away from this potential homeland, it decreases. So the Balkan Peninsula, we have a clear gradient from more to less. In Germany, there's a big barrier. Basically, in Western Germany, you have nothing. There's one core population that has quite a lot, that are the so-called Sorbs. They are Slavic-speaking minority that persisted there since the 10th century, basically. And basically, Eastern Germans from Leipzig, where my institute is, they have roughly like 40% of Slavic-related ancestry. And then the same pattern as with the Balkan Peninsula, we see basically to the east with this, for example, with Moldovians and other Finno-Ugric populations in Russia, that the more east you come, the less Slavic period ancestry they have. And we also measure like certain amounts in the Baltics that might be to some backflow into these areas. So how can new data help? I hope I was able to show that there are still sampling gaps. And it was very nice that we could start a cooperation with your team here, and we're actually able to produce some new data and from several sites at the borders between Lithuania and Belarus. And this is, of course, very interesting because it's very much unknown territory during the Iron Age migration period and the early Middle Ages. And this is basically a project I'm preparing since like 1 year, where I've collected much new data from central Germany, from the Baltic coast area of Germany. We will have material from southern Sweden, from Denmark, and hopefully much more material from Poland, as well as material from Ukraine. And it will be very interesting to see how these groups from southern Lithuania have interacted with their neighbors. Not only in regard to the question how this gene pool of the early Slavs has formed, but also in later time periods. And just want to show, to finish this presentation, some preliminary results. Short comment, you also got some, that's very, very cool, also some material from Belarus. This would be the first material from Belarus that is actually published, if we publish it soon, I guess, because Belarus is really, there's not a single genome from this area. And what you can see here is in purple, we have the Lithuanian samples, and in blue, the Belarusian samples, where you see they plot pretty much on top of present-day Finns, Estonians, and Latvians, and a little bit different from present-day Lithuanians. So there's a little bit of a shift indicative that the present-day Lithuanian population has a little bit more like Western and Southern European ancestry compared to the early medieval and Iron Age population. As many populations, actually, because due to increased mobility and contacts, of course, there's a lot of admixture, and the extremes of clients get more pulled inside the direction of the center, which would be here. And then it's very interesting, the Belarusian samples pretty much, at least for the early Middle Ages and then for the modern period, plot very similar to Southern Lithuanians and Belarusians and Polish, whereas the Iron Age Belarusians look genetically very much like Lithuanians. I mean, since they were sampled close to the border of Lithuania, of course, it makes sense. They're not from the south of Belarus, but it's interesting because it shows that this Baltic Iron Age gene pool has extended further south, and probably much further south, but we don't know without material from Belarus. If we reconstruct some close pedigrees and kinship again, it was very interesting to see that we find many relatives within sites. That's not so rare, that happens. But what is very interesting is that these are now each is an individual, and the first 3 letters are basically the sites. It's an internal documentation system for the sites. It's not important here which sites they are, but what you see here is that for these red cells, the individuals come from different sites. And that's fascinating, because in the archaeogenetic record, relatives between sites, close genetic relatives, that's important. So first, second, third degree are very rare, even from sites that are just 10 kilometers apart from each other and from the same time period. We nearly never find close relatives. So the fact that we find so many inter-site close relatives between these Lithuanian sites already makes some interesting makes for some interesting hypothesis, maybe about like the mobility between sites, maybe something about population sizes. It's really interesting. So that's already pretty cool. If we look at the distant relatives, so larger than 4th degree, we find even more of these inter-site pairs. So that is more reflective of less of like mobility, but really of population size. That's no secret. Northern Europeans have much smaller effective population sizes than Southern Europeans, like Scandinavians, people from the Baltics, Finns. Their effective population sizes are so much smaller than people from Italy or Iberia. That's simply just a geographical pattern. However, we also find very interesting, unexpected distant relatives. For example, we find between this Belarusian individual, we find connections to Viking period individuals from Gothland. So this is, of course, very cool because we know that at some point Scandinavians protruded into the territory of Belarus and Russia and formed these Rus territories there. So maybe we see some traces here. However, the most interesting connection that I found most fascinating was the connection to a Roman period sample from Poland, a 6th or 7th degree relative. That's very interesting because this individual, it's this square here, you see he or she falls a little bit distant from the core Lithuanian group. So he or she has some ancestry that is not from Iron Age or early medieval Lithuania. It's shifted into the direction of wheelbark culture individuals from Poland. And as I say, they plot pretty much with Scandinavians. And the individual, it's a 6th degree relative to this blue square, is also shifted, but shifted from the Poland. So this Poland wheelbark culture individual is shifted towards the Lithuanians. So if we do the ancestry decomposition, we can measure that this individual has 44% Lithuanian Iron Age derived ancestry, and our Lithuanian individual has 18% Swedish Iron Age ancestry, which is also the ancestry that we find in Poland wheelbark individuals. So basically what we can trace here is a two-step process of genetic relationship and mingling reproduction between like the wheelbark culture individuals in Poland and their geographically close neighbors in Lithuania. And probably, I mean, we don't have individuals from Kaliningrad, we could probably assume that there was an interaction zone along the Baltic coast between like this presumably Germanic Gothic tribes and the Baltic tribes inhabiting the areas further to the east. And we know from archaeology, of course, there are graves that have, for example, different elements. As far as I heard, the wheelbark culture is actually very influenced by Western Baltic cultures, and yeah, and vice versa. And of course, the amber trade routes would be a very good plausible explanation for such context, besides the close geographical proximity. But I think that's very interesting that this genetically, in a European context, seen differentiated populations have these interactions. And as you see, there are actually multiple more individuals here that are a little bit shifted into this direction. So it's potentially not just a singular event. There are several individuals that have Sweden or Poland wheelbark culture related ancestry. So potentially that was not such a rare occurrence, but they were good neighbors. And on the other hand, we also find several individuals that are distant relatives to Lithuanian Iron Age individuals everywhere across like East Central Europe. So what we clearly see, there was not only connections to the northwest, but also to the south. And that's maybe one of the second last point before I finish my presentation. This is a very interesting observation. It's maybe a little bit nerdy, but I want to highlight here anyway, that it also tells us something that we maybe cannot even see here in the present day genetic makeup of Europe. Because in the present day genetic makeup, we see that the Balkan Peninsula is organized as a cline from basically northeastern European populations to southern European populations. And as you maybe remember, I've shown that basically the Iron Age and Roman period populations of the northern Balkans plotted here and of the southern Balkans plotted here. So basically what happened is more southern ancestry came in. Most of the Balkan populations during the Roman period fell like here. Then the Slavic ancestry or northeastern European derived ancestry entered and basically formed this admixture cline. However, before this admixture cline existed, we find in Bronze Age and early Iron Age populations from Albania and Romania an admixture cline that is no longer visible in the ancient DNA data. It starts here in Lithuania, and probably Latvia and Estonia would be also here, goes over Belarus, Eastern Poland, then I have not plotted it here because it would be a little bit too overcrowded, would be samples from Moldova and Hungary, and then we see here Romania and Albania. So maybe there was a Paleo-Balkan Baltics cline that is basically an interaction zone between populations from the Baltic coast area and the Balkan Peninsula. That's basically a genetic diversity that does not longer exist. All here, this area in the present day PCA space is empty. It's the Adriatic Sea. There's no admixture. But presumably, just maybe 2,500, 3,000 years ago, there was quite a vivid population genetic diversity through Bulgaria, Eastern Romania, Moldova, and Ukraine that connected these genetic diverse ancestries in an admixture climb. And that's simply lost today. And to end this presentation, to just come back to my case study and how new data can help, this is just, it's a complicated plot, but I just want to highlight one main message here. Basically, this is a metric plot where I test for two-way admixture models how well they explain the early Slavic gene pool that we observe in Poland. And the legend is easy. The redder the color, the worse, the most horrible is the model. So 0.01 is a horrible, horrible model. That's probably not the truth. On the other hand, blue is probably also not the truth, but it comes closer to the truth. So what we see is that this combination of these two groups here produces blue models, so produces good fits. So probably this is closer to the truth. And I've marked the 3 best fitting models of this hundreds of models that are depicted here. And the main message here is to explain the early Slavic gene pool, we need a southern source. The southern source can be from Hungary, can be from Slovakia, can be from Moldova, can be from different parts of the Balkan Peninsula. It actually does not matter. However, what we always see is basically that the ancestry that best approximates the northeastern European ancestry is Iron Age Lithuanian genetic information. So that does not, of course, not mean that Iron Age individuals from Lithuania migrated south and formed this early Slavic gene pool. That's much too late. But individuals that were closely related to the Iron Age gene pool of Lithuania were probably the groups that expanded southwards and mixed with Balkan-related populations to form the early Slavic gene pool. So that shows really that there's more need for sampling, that we understand which groups are how related to each other. Otherwise, we have to work with representatives, with proxies. But it clearly shows that the Baltic groups currently are very understudied, but they are very important for the population genetic history of large parts of Europe. And I hope that with the development of this institute here and our further cooperation, we can shed some light on this very interesting demographic trajectories here and elsewhere in Europe. And with that, I thank you very much for your attention.
Thank you so much, Joscha. It was amazing. And perhaps now there is a time for questions. Someone in the audience would like to ask something?
Lord, okay. Okay, thank you. This is mainly out of curiosity regarding the genetic diversity using mitochondrial DNA markers. I wanted to ask if conservation rates, or in other words, mutation rates, were accounted when evaluating the genetic diversity, because I'm assuming the genetic markers were assessed using current mitochondrial data.
Yeah, so basically for the mtDNA is normally quite well preserved in ancient DNA samples. So that's both good and bad, because the mtDNA is, of course, in most cases less informative on the fine-scale population genetics. But otherwise, it's often 200 times more better preserved than the autosomal DNA. So what we can often do is basically reconstruct basically a simple de novo mapping. So it's not so horrible regarding the actual classification of mtDNA haplogroups. What we definitely can say is that this is not an effect of some kind of an underestimation of haplotype diversity that exists clearly also in the Y chromosomes. Actually, I've seen there are much more issues that actually many of the Y chromosomes that are, it's mostly from the Neolithic, but also from later times, are not well represented with the present day data banks because they do not exist or are super rare. I think like a good example is this a haplogroup from West Africa that was identified in an Afro-American male from the US, which is one of the deepest splits in human Y chromosome diversity. So it's extremely interesting from a human evolution perspective and was simply not found because it's so rare. So we have seen that Y chromosome lineages can go undetected, but we can clearly, even if we just do not know the exact haplotype of the mtDNA haplogroup, we can just see that there are so many macro haplotypes in comparison to the Y chromosome haplotypes that there's a massive difference. It's clearly not just a bias. If that answers your question.
It does. Thank you.
Thank you.
A follow-up question, a little bit unrelated, but again, out of curiosity, are there any other genetic diversity markers that are used as a standard in paleogenomics?
That depends, of course, what exactly you want to measure for diversity. Also, a good measurement of diversity is, for example, the pairwise mismatch rate that is, for example, used for the calculation of close relatives. That's normally like 25% for unrelated individuals and can go up if it's like from different populations, and then you would have like 19% if you're first degree relatives. So that's a measurement you can do. You can use this F3 statistics, this outgroup F3 statistics that measure shared genetic drift. That's normally in, that's also a measurement of genetic diversity. For example, early Bronze Age populations have much lower much higher levels of shared genetic drift. Present-day populations have less. You can, of course, also use, I've not shown this here, runs of homozygosity. That's very similar to ABD segments. So these are stretches of DNA that are homozygous and not heterozygous because you inherit them from your parents or from ancestors that are closely related. So basically, they're a mark of consanguinity. And based on those, you can identify if somebody is basically the product of consanguinity, first degree, second degree, third degree cousins, for example. Horrible cases where parent offspring even exist in the ancient DNA context, or siblings, but it's very, very rare. But it can also be used for effective population size. For example, we did this for the Slavic populations, Slavic period populations. Using runs of homozygosity, one can estimate effective population sizes between 10 and 20,000 individuals. This is for runs of homozygosity quite expected. Like everything over 6,000 is quite a lot, and it's measured using. That's maybe too much into detail. And you can use, of course, identity by descent segments, and these probably represent the actual census population sizes a little bit better than the effective population sizes based on row. With that, we can see that, for example, in Ukraine, we have like 50, 60,000 individuals, and in Eastern Germany, like as well. So they measure different types of diversity, but all of them are related and can be inferred from ancient DNA.
Thank you. Very different from modern genetic diversity studies.
Two questions which may be stupid. You know, I'm an archaeologist. I have to process many things in my head before I, yeah, you know, some things seem right the opposite than I thought as an archaeologist. Can you turn several slides back? Yes, let's say those arrows. Mm-hmm. If you draw the arrows right the opposite way, it would be easier for me as an archaeologist to understand that. How do you, let's say, know the direction of the arrows? Did those particular individuals have their ancestors in the local places? Yeah. Let's say the one from, I guess, from Planck and the one from Wielborg.
Mm-hmm, mm-hmm, mm-hmm. So for this arrow, I would say the other direction is, well, you could put it also the other way around. It makes no difference in this case. For these arrows, I would say these are basically a little bit for visualization. I would say this arrow is in this direction because they share quite a lot of IBD fragments with this individual. So this individual is clearly out of the context of the Lithuanian Iron Age population. And he or she shows then this one connection to this individual. And this connection also shows one connection in this direction. So basically it should be maybe an arrow with two arrow heads. So I'm going in this direction. While this individual it's from a Polish wheelbark culture site. and it shows also many IBD connections to the other Polish Biber culture individuals. So it's also clearly an individual, although it receives quite a lot, like nearly 50% of its ancestry, it's more connected with this population. But I see your point here. It's clear that this is a matter of visualization. And if you're also right, actually this individual with like 50% of its ancestry coming from this source and this source, it's actually neither belonging to the core group of both of them, but it's actually like a recent mixture. It's both groups.
Okay, and a very simple question. When you are speaking about second level of connection, how do you call it? Can you precisely tell between a cousin and a grandson, or is it genetically the same?
So yes, so first degree is very easy. So if your siblings are parent offspring, that's very easy.
Yeah, it is.
For second degrees, it's getting more difficult. It's theoretically possible from the IBD sharing to infer that to some extent. We can differentiate between for example, half siblings, uncle, nephew, niece, aunt, and grandchildren, grandparent. That's possible, but depends on the quality of the data, because these methods need quite a lot of these diplogenotypes to. It's all probability. It's all like percentages. So in the end, you will get an estimate, but maybe it has like very large standard errors, and then you cannot say anything. But theoretically, it's possible to differentiate that. In most cases, if you have like a large pedigree, then it's much easier to just do it from the pedigree. If you see this individual, we can simply, I have to jump back a little bit here. If you have reconstructed like already all these relationships, then some of them are mutually exclusive. If you're like, if these, if you know these must be, these are not related, but those are first degree relatives to each other, and they have the same mtDNA haplogroup, and which is the same like this one, and they are not related, then we know the direction must be, these must be the parents, and these must be both children, and must be siblings. And then, for example, if you know this is in second degree individual, and we have then here this part of the family, you can like from the reconstruction already say this must be then the grandmother and these must be the grandchildren, because then the possibility that this is like, I don't know, an aunt or a cousin is then not longer possible. So the different connections between the individuals help to exclude some of the possibilities. And this is often much more precise than actually what the single tool can tell you. Like this is more like avuncular or, I don't know, grandparent, grandson.
Thanks.
Do we have any more questions? Oh, Jurgis, please.
Thank you very much for a very interesting talk. We already discussed this a bit earlier. As a linguist, I'm really looking forward to the inclusion of ancient DNA from the Western Baltic populations, the Yetvingians and Old Prussians, because currently we see the Eastern Baltic data. They are ancient and also modern that are playing in the picture, but it will be really interesting to see what happens when we add the Yetvingian and Old Prussian data into the picture, both in terms, I mean, of the ethnogenesis of the Balts, but also in their relation to the Slavic splits and the Balto-Slavic proto-history.
Yeah, absolutely. I can just agree. As I highlighted here, there's simply some areas are now, also thanks to the HistoGenes project, just very well oversampled. Like Pannonia with 7,000 samples has nearly equal as many samples as like all of Iberia and Italy together, and like England is the same case. Like for England, from London, there's a large project that will also sequence like thousands of individuals. So there will be regions that are vastly over-analyzed, while other regions, due to many different factors, are under-analyzed. And for example, as the area you have spoken of, you see there are some samples, but they are all very young. These are mostly migration period, Slavic period samples, but we don't have like any Bronze Age samples. We have some older samples than here from a little bit to the south that we can already maybe have a look. But like also from the Baltic area, we have the issue that there's like from Lithuania and Latvia, there are like 5 and 6 Bronze Age individuals from the late Bronze Age, but nearly nothing from published currently from the Iron Age. So I think our data will be very valuable for also many other studies, and I think also like for future studies, I think these are time periods that should be targeted because they could be very interested for researchers that both study, like, of course, the connections to the west, but of course also the connections to the north. I mean, in Gothland, for example, during the Viking period, I think like 40% of the ancestry is Baltics derived. So there's clearly a lot of individual mobility going on. So this is, of course, also very interesting from a social standpoint. So I think also Baltic data could be very interesting in elucidating these questions. Yeah, I think only more sampling can help.
Hello, thank you. I was wondering what the ratio on your team is from geneticist to social scientist to archaeologist when you're working out your results of the data.
This is now a very good question. I fear I cannot give a good answer for that. I mean, historians are clearly in the PI section overrepresented. There are 2 historians and only 1 archaeologist and 1 geneticist. No woman, which is, I guess, a little bit of a problem. But I think on the second level, there's then quite a lot. Like my immediate supervisor, Susanna Hoffmanova, is really involved here. So I hope it does not look too bad for the outside. I would say we have, I would say, nearly as many archaeologists and anthropologists as geneticists. Historians, it's a little bit less, but of course, historians provide a little bit of a different input, so having the same number would not be like necessary. Not because I don't like historians, but like because of course there are only so many historical sources that are like commenting on like the areas we're analyzing. So of course you don't need 50 of them. But anthropologists we have, especially from Vienna and from Budapest, there's quite a few, and then archaeologists. I mean, of course, the actual core group of people that analyze data is, of course, also then significantly different from the people that just provide context information that are less than more involved. So I would say if we're just talking about genetics, then we're probably also just talking about like maybe 20 people that are actively analyzing most of the material. And from the archaeologists, it's probably also like 15 or something like this, plus 5 anthropologists, 6 anthropologists. But of course, then there's like large amounts of people that are more in the background that are also extremely important, like all the lab technicians, for example, that neither fall into like the analyzing group nor in the like supervision group, but of course they provide most of the necessary work. You cannot analyze any genome if it's not produced in the laboratory. And of course, they're in Brno, they're in Germany. Some of them are also in New York. So it's a quite large team, but I think it's very balanced in terms of the different disciplines. And yeah, maybe also a short comment. I think it's also very important that these groups also work very tightly together. So each year there's a major retreat where all people involved in the project come together, and then there are presentations and project discussions. And the idea is that for all the major genetics papers, there's, besides the genetics co-author, always an archaeology or history co-author, like on the first 2 positions. And that for nearly all genetic publications, we also have an archaeology publication where then basically the archaeologist can be then the first author and provide the data in the style that is required by the archaeology or anthropology community. So we try that it's all roughly equally distributed and everybody gets their fair share of the data wealth that is produced.
One question online, and Mantas Dobras is asking, is the raw DNA data produced during HistoGenes project available online, or will it be?
Yeah, so all data that is produced will be publicly available on the normal ancient DNA data banks. Of course, it must be published beforehand. That's the important thing. So the two major papers I've shown, and there's another, I think, current biology paper about Longobards migrations to Pannonia. They are published, and so all the data is available. The BAM files are available. In our team, we always also want to publish the FASTQ files so everybody can do their own mapping. And we have also started to provide so-called Poseidon packages. Poseidon is a repository for genotype data, so that you don't have to genotype the BAM files yourself, but you can actually just download Eigenstratt or Plink file, and you can directly start doing your PCAs right away. That's basically our concept to distribute that also in a more easy way. So everything will be available.
Thank you. That's a good news. Any more comments, questions? Yeah, so probably big round of applause.
Thank you very much.